Good day, this is the Professor, CBMD PhD. And today's Friday, August 5th, 2022. It is 4:40 and 27 seconds, a.m. Eastern Standard Time. And today's research, bladder cancer. So let's hop to it here. The genetics may predict bladder cancer immunotherapy response. Date August 4th, 2022. Source, Cedars-Sinai Medical Center. Investigators have identified genetic signatures that could predict whether tumors in patients with bladder and other cancers will respond to immunotherapy. The work indicates that these genetic signatures may prove to be tremendously valuable in predicting immunotherapy response in patients with bladder cancer, but also other tumor types. The researchers will continue investigating these biomarkers with the goal of bringing them into clinical use and improving patient outcomes. During the past 5 years, anti-PD-1, PD-L1 therapy, a type of cancer immunotherapy that paves the way for the body's immune system to attack tumor cells, has proved effective against many cancer types. It has proven very effective against melanoma and revolutionized lung cancer treatment. Bladder cancer is considered one of the more responsive tumor types, but still has just a 25% durable response rate, so improvement is still needed. When a tumor causes a host immune reaction, immune cells usually are able to infiltrate the core of the tumor, and scientists call it a hot tumor. But some tumors, on the other hand, block immune cells from infiltrating and are known as cold tumors. A previous study implicated a gene called discoiding domain receptor tyrosine 2 in contributing to anti-PD-1 resistance in animal models and several tumor types. In collaboration between cancer biologists and bioinformatics researchers, the new study further investigated the discoiding domain receptor gene family using human cancer datasets and various tumor types. The study analyzed data from the Cancer Genome Atlas program, a publicly available database with information on hundreds of cancer samples, and to look how expression of discoiding domain receptor 2, a related gene, discoiding domain receptor 1, correlated with a host immune response to a tumor using bladder cancer as a model. The researchers also looked at how genes regulated by discoiding domain receptor 2 and discoiding domain receptor 1. Important example, gene signature, correlated. Researchers then looked at patented data, a clinical trial that evaluated immunotherapy response in bladder cancer to see if these responses correlated with discoiding domain receptor 2 and discoiding domain receptor 1 expression on their related gene signatures. While the discoiding domain receptor 1 and discoiding domain receptor 2 genes are members of the same family, they have very different effects on tumors. The tumors with high expression of discoiding domain receptor 1 tend to exhibit low expression of discoiding domain receptor 2, and vice versa, with high discoiding domain receptor 1 tumors are cold, while high discoiding domain receptor 2 tumors are hot. Investigators also identified 4 unique gene signatures modulated by discoiding domain receptor 1 and discoiding domain receptor 2 that were closely associated with tumor response to immunotherapy. They checked these genetic signatures in several additional sets of publicly available patient data across multiple cancer types. Researchers found that these gene signatures were clearly associated with response to immunotherapy in bladder cancer and lung cancer tumors in multiple patient groups. The researchers also evaluated the signature in publicly available data on melanoma, glioblastoma, and blood cancers with similar results. The next step is to validate these signatures in a prospective clinical trial. This could yield new tools that allow clinicians to determine pretreatment whether given patients will likely respond to anti-PD-1, PD-L1 therapy. They can then proceed with the anti-PD-1, PD-L1 therapy for those patients who will derive the most benefit and offer alternative therapies for patients not likely to respond, improving outcomes for all. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.